about my work in decoding calcium signals. In particular, I'm interested in studying the role that luminal calcium plays in these signals. So, as we've seen yesterday and today, and Silvina already said, calcium signals are a universal mechanism that cells have to change information between cells and also inside the cells. And the message encoded by calcium is in its spatial-temporal distribution. So signals occur when calcium is released from internal reservoirs, as are the sarcoplasmic or endoplasmic reticulum, and calcium exits through channels. The two main channels are the ryanodine receptor and the IP3 receptor. We are interested in studying IP3 receptor-mediated signal. So this channel opens when it binds IP3 and calcium. But the role that calcium has on this channel is dual. At a low cytosolic calcium concentration, it activates the receptor, and at high concentrations, it inhibits the receptors. So in this way, calcium modulates its own release. And also the interplay between the cytosolic calcium concentration and the receptor distribution in the ER membrane can determine the kind of signal that we can have. And also, as Silvina already said, we have a wide variety of signals, from very localized and brief ones to global signals that can last for several hours, depending on the cell type. So, as I already told you, the role that cytosolic calcium plays in this IP3R-mediated signal is very well known and studied, but the role that luminal calcium plays is still unclear. Luminal calcium is calcium inside the reservoir, inside the endoplasmic reticulum. So the aim of my work is trying to understand what is the role of this luminal calcium in this IP3R-mediated signals. So first of all, I would like to say that we work in these cells, in Xenopus oocytes, and this is a particularly good model to study IP3R-mediated signals because it's the only receptor present in these cells. And also for us as physicists doing experiments, it's particularly easy to work with these cells because they are huge. So they are really easy to manipulate. So the way in which we study signals is that we inject fluorescent dyes and cage IP3, and then by photoreleasing IP3, we have a way of inducing signals, and by looking at the fluorescence with confocal microscopy, we can have information about cytosolic calcium. So, to the best of our knowledge, there has been a lot of studies about cytosolic calcium with cytosolic dyes, especially fluo4 and also Rod2, but in these cells, we hadn't found studies about luminal calcium. So what we propose is a protocol to label simultaneously luminal calcium with this dye, with fluo5N, and with Rod2 with the cytosolic dye. So in this way, we can see signals from both the inside and the outside of the reservoir. So the first things I did were some control experiments to make sure that this dye was effectively entering the lumen and labeling luminal calcium, and that it was not staying in the cytosol. So the first experiment that I'm going to show you is just a static imaging experiment. So in the upper panel, you can see signals from, it's an imaging experiment from inside the oocyte. This is fluo4, a cytosolic dye, injected simultaneously with Rod2, another cytosolic dye, and in the superimposed image, you can see that they colocalize pretty well. So this is this is like kind of trivial because we know that they are both cytosolic dyes, but this is to compare with the lower panel. In the lower panel, in channel 1, we have fluo5 signal, in channel 2, Rod2 signal, and in the superimposed image, you can see that both images not only do not colocalize, but they seem to be kind of complementary. So in this was our first control to see that fluo5 was not staying in the cytosol, and it was labeling calcium inside some reservoir. So the following experiment that we did is that we injected fluo5 along with the membrane tracer, DI, and simultaneously, and we observed both signals, and another time we can see that they don't colocalize, so the luminal dye is not staying in the cytosol. It's not labeling the membrane, but it seems to be labeling a structure that is bounded by the ER membrane. So now we can say that we're sure that fluo5 is effectively entering the ER lumen and labeling luminal calcium. And also with these experiments, we have a way of looking at the shape that this reservoir has, because we're looking at its membrane and its interior. So what we did then were some signaling experiments with this luminal dye and with our cytosolic dye. So a couple of years ago, I did this experiment. This is like a cartoon of the experiment, where I injected Rod2, the cytosolic dye, fluo5, and KJP3. So before the UV light, this is like the basal situation, Rod2 signal should be low because the calcium basal concentration in the cytosol is relatively low, and the fluo5 signal should be high. And then after the UV light, as IP3 is photorelease and binds to the the channels open and calcium flows from the ER into the cytosol. So this is the cartoon, and this is what we measure. These are mean fluorescent traces from a region inside the oocyte, and as I told you, the signal from the cytosolic dye increases and then goes back, but the signal from the luminal dye that we observed decreased, but not as much as the cytosolic dye signal. So we were expecting like mirrored signal, because all the calcium that's leaving the ER is entering the cytosol. So at that time, we couldn't explain why the difference in the rates for these images. So last year I did some new experiments, but just using the luminal dye with the aim of characterizing luminal calcium dynamics. So we did like more global signals. You cannot see, maybe you can see here the time scale is in minutes, and the time scale before was in seconds. So these are more like global signals. This violet bar is the UV light, so it's our stimulus, and it's more powerful and longer than the previous stimulus. So now what we want is kind of, we want to deplete the ER, or try to deplete the ER and have a way to quantify the behavior of luminal calcium during this global signal. So as we expected, after the stimulus, the signal drops, indicating that calcium is leaving the ER, and then when the UV light is turned off, the signal starts to recover in a slower way than it was released. So we did many of these experiments for a lot of cells and for different regions in the cells. We took those averages, we did some statistics, and we wanted to obtain release rates for these parts and recovery rates. And we wanted to attribute these two things to two different processes. The release rate, we thought that it was just associated with a calcium current through the IP3 receptors, like for a calcium flux through the receptors. And this part we attributed it to the reuptake of calcium inside the ER by the sarcopamus in its membrane. So we built a very simple model to go from fluorescence to underlying calcium concentration. Basically, we said that that calcium-bound dye represents a fraction of total calcium in the lumen, and that the fluorescence is proportional to calcium-bound dye concentration. In this way, we can go from our experiments and our experimental rates of change for fluorescence to rates of change for the luminal calcium. And as I told you before, we attributed this to just calcium flux to the IP3R, and this part to a calcium flux that was entering the ER to the sarcoplasm. And the numbers we obtained for this flux were very good. We compared them with other estimations for this flux for other cells, and we were happy with this number. But then when we compared our experimental estimations for these rates and for this, we found that the rate that we obtained experimentally was much smaller than the one that we expected if we took into account the number of receptors in the area that we observed. And then we were stuck with this question for a long time, and what we propose is that during the release, the AP3R are not the only mechanisms involved, but in our cells there is another calcium flux that is entering in this game in the lumen, that is the calcium flux that is being released from intraluminal buffers. because we knew that most of calcium inside the ER is being buffered by proteins like calreticulin. So we proposed that the reason why we cannot see the bigger release that we expected is that we have a very, very fast release of calcium from the luminal buffer, and that this release is preventing us from seeing this decrease in more brief experiments. So going back to the previous experiment, we think that this is the reason why for us we cannot see mirror signals, because we have this very fast calcium release mechanism inside the luminal that is preventing calcium depletion, and in this way we cannot see the decrease that they see in other cell types like this muscle cell. And another argument in favor of our hypothesis is that in our cells the volume occupied by the ER is considerably large. So if we compare our surface volume ratios for our cells with the one from other cells, you can also see why the considerable volume of this structure contributes in favor for this fast refilling of calcium. So we were able to label luminal and cytosolic calcium simultaneously, and also to observe small signals and global signals. We were also able to quantify calcium fluxes, luminal calcium fluxes for these signals, and with a very, very simple model, we obtained parameters that fit our data and are also comparable with other theoretical parameters, and also we link the spatial aspect of the signaling, that is the shape of the reservoir and the dynamic of calcium inside the ER. So I would like to thank the organizers for inviting us, Silvina, the whole group, and everybody for staying till this late and listening to us. Thank you.
So is there anything special about oocytes for this double labeling? Could you do it in cardiac cells? Will the dye go through the SR?
Are you asking why I use oocytes?
No, I know why you use oocytes, because they're easier, but in some sense the most useful application might be to cardiac cells.
Yeah.
So I'm just wondering if the same dye technique will go through the SR.
It already works in cardiac cells. We took inspiration in cardiac cell experiments, and we wanted to try this dye for ours.
Okay.
No,
No, that's what I'm saying. They do two. That's what I'm asking about.
Yeah.
Not the two dyes.
We do it in two dyes, yes, also.
Okay, so the two dye thing should work also in cardiac cells.
Yeah.
Right, okay.
Oh, so in your initial images, you showed the cytosolic, and then you had a lot of these voids, but only some of those showed the luminal, binding. So are those other voids, or those
dark spots?
ER without the calcium in them? Why do you see this heterogeneity?
These dark spots are granules.
Pardon?
Are granules. There are structures inside the oocyte that are dark, and you don't have calcium inside them, and calcium dyes don't enter those spots.
Right, so you see then uniform calcium throughout all the ER. There's no compartmentalization, because these other dark spots are granules.
The dark spots are granules, and it's not homogeneous. Are you asking about the distribution?
Well, the green, is the concentration equal across all of the ER?
No, it's very heterogeneous, because also buffers are not distributed homogeneously inside the ER, and we're doing another work study.
Does that have importance, the fact that you get this pretty wide heterogeneity within the ER?
That I have a very wide?
That you get this pretty dramatic heterogeneity within the ER?
Yes,
luminal? Is that important?
Yes, it's very important for signal initiation, but we're looking at this spatial aspect and heterogeneity and studying how that can affect the cytosolic signals.
Oh, great, nice.
One thing that we saw is when the release starts, it gets more homogeneous. So communication, and it's pretty, well, it's not long distance, but it gets
Thank
which
you.
is something that we are studying.